Today we're going to talk about signal pathways, which include inositol triphosphate. The beginning of the pathway occurs on the cell membrane, specifically on the G protein receptor, also known as GPCR. It's a transmembrane protein and common receptor in eukaryotic cells. It's called the transmembrane protein because it has 7 transmembrane segments. To start this process, the ligand, or signaling molecule, non-covalently binds to the G protein. The activated receptor changes its conformation and becomes ready to bind with the G protein. The G protein is a lipid-anchored protein divided into alpha, beta, and gamma subunits. So back to the receptor. Activated receptor couples with G protein, causes G protein alpha subunit to exchange existing GDP to GTP. When the G protein is activated, it causes this alpha subunit and the GTP to separate from the beta and gamma subunits. The separated G protein alpha subunit combines to the phospholipase C, also known as PLC, and activates the PLC, which is an enzyme that digests phospholipids in the cell membrane. PLC uses energy to break the bond between the head and tail group of the phospholipid called IP3. You guys might better understand if you see the chemical structure of PIP2. PIP2, like other phospholipids, has 2 lipid chains bind to glycerol, and a head group which has inositol with 2 phosphates. Phospholipase C breaks bonds between phosphorus and oxygen to break the tail and head group to make inositol triphosphate, also known as IP3, and diaxyglycerol, also known as DAG, which are the second messengers. IP3 is released from the cell membrane, diffuses in cytosol, then binds to the ER membrane calcium ion channel. The open channels release calcium ions into cytosol. Calcium ions release from the ER and bind with PKC. The main advantage of a second messenger is amplification of signals. If there were no second messengers, one ligand could only give one signal. But you saw that one signal molecule released several IP3s, which released a lot more calcium ions. Calcium ions transmit various signals, so using secondary messengers is more efficient.